The overall goal of the cell-based assay is to identify molecules that bind to a G protein-coupled receptor based on their capability to compete with a fluorescently labeled ligand for binding to the receptor.
Competition binding assays can support G protein-coupled receptor drug discovery, as we will demonstrate with this protocol for the chemokine receptor CXCR4. The main advantage of this method is that living cells are used to analyze receptor binding instead of membrane preparations, and that the use of radioactively labeled probes is avoided.
Though this method has initially been set up to study binding to the chemokine receptor CXCR4, it can also be developed for other GPCRs for which fluorescently labeled ligands exist.
To begin, dispense 100 microliters of compound solution into a clear, 96-well round bottom plate according to a predefined experimental layout. Add 50 microliters of cell suspension from a reagent reservoir into the 96-well plate using a multi-channel pipette. Incubate the plate for 15 minutes at room temperature in the dark. Following incubation, add 50 microliters of fluorescently labeled 100 nanogram per milliliter CXCl12 from a similar reagent reservoir to the wells of the 96-well plate. Incubate for 30 minutes at room temperature in the dark. Next, centrifuge the 96